It's really a pleasure to introduce Suzie. She has been the recipient of many, many awards. She got a Career Award. She got a Picasso Award that took her to the White House, just like David did. She was named one of the TR 100 Young Investigator. She just recently got a Cancer Alliance Award. And she's been doing some fantastic job in terms of drug delivery and different modalities, and in particular going across the brain-blood barrier. And we're going to hear all about this today, and it's really a pleasure to have you telling us about molecular engineering of these systems. Suzie?
Francois. So it's a great pleasure to be here to celebrate this institute. My laboratory at the University of Washington focuses on drug delivery. So today I will be telling you about some of the materials that we've been engineering, trying to make delivery systems that are specific, programmable, and also efficient. So what I'm showing here on this first slide are some examples of drug delivery that might be familiar to you. So up here is a long-lasting medication from Pfizer that's used to treat high blood pressure. The first iteration of this that came out, patients had to take 3 times a day. And with this long-acting version, it's an osmotic pump that releases over a prolonged period of time, so you only have to take it once a day. The second one you might be familiar with is the nicotine patch, where you can just put it on your arm and you can get small doses of nicotine. The third one here is an aerosol that is used to treat asthma, and so that gets the anti-inflammatory drug into the lungs very quickly. And then this last example here is a formulation used to treat cancer. Doxorubicin is a chemotherapeutic drug that is quite effective, but it's also very toxic, and there are a lot of side effects with it. So with this delivery system, the doxorubicin is encapsulated into a fat bubble, which allows it to be dosed at a lower dosage level, and also this formulation has less heart toxicity. So one of the things to note in the majority of current drug delivery technologies is that these are really methods to take current drugs that are already available and make them more convenient, maybe more safe, and improve patient compliance. So most of these drugs that I'm talking about, that I've given up here as an examples, these are all what are called small molecule drugs. So here I'm showing the structure of nifedipine, which is the active ingredient in this procardia that I talked about. And if you take an average of how big it is, it's about 1 nanometer in diameter. The drugs that Seattle Genetics, so Eric Dobmeier represents this company who was on this, the lunch panel, is synthesizing antibody drug conjugates, and these are on the order of about 10 nanometers in diameter. So they're 10 times larger. And these antibody-based drugs, these protein-based drugs, currently act on extracellular targets, in part because of the size restriction. So these things are small enough that they can get inside your cells and go quite a bit throughout your body. These antibodies are more restricted in their diffusion, or in the places that they can go in your body. And then one of the drugs that I'll talk about today are nucleic acid-based drugs. And this is a plasmid that has a gene for gene delivery. And now this one is about 1,000 nanometers. And so we're looking at something that is 1,000 times bigger than most of these small molecules. So these classes of drugs are now called macromolecules or biologics. And there's a relatively large group of us here at the University of Washington that's working under the leadership of Pat Staten in the Center for Intracellular Biologics Delivery to deliver these large-sized molecules. And so in this case, these types of molecules where we're trying to get proteins inside the cell or get nucleic acids inside the cell, the drug delivery technology becomes, in my mind, just as important as the drug itself. Because without the drug delivery technology, the drug actually cannot be translated to the clinic because the drug cannot get to its site of action. And so today I'll touch on some of the technologies that we're developing in our laboratory. I talked quite a bit about this intracellular delivery of macromolecules. I hope you can appreciate that these large molecules need something to help escort them inside the cell. I'll also talk a little bit about what are considered undruggable targets. So I'll talk about delivery to the brain. And a lot of the diseases that are associated with the brain and the spinal cord are considered untreatable because it's very difficult to get drugs into that system. And I'll give some examples of this last topic of preferential delivery to desired cells. This is particularly important for cancer treatment, where most of the drugs are out to kill cells, and becomes very important to only specifically kill the cancer cells and not the other cells that are in the body. So I will talk about the molecular engineering of vehicles, and I really appreciated Matt's definition of molecular engineering. And I hope that I can convey that in our laboratory we are combining molecular biology and chemistry technologies to develop delivery systems that we hope to be able to translate clinically. So the first example I'll give is gene delivery to neurons, and we're specifically interested in delivering to the brain. And in that case, I'll highlight a material that we've designed to have programmed intracellular delivery so that the material is stably encapsulated when it's outside of the cell. But once it goes inside the cell, it'll be triggered to release its payload. And then in the second example, I'll talk about two projects we have for tumor-targeted delivery. And here I'll address the idea of specificity. So how can we get materials more specifically to tumors and less to normal cells? So for the first topic, so gene delivery to neurons, again, we're interested in delivering genes to the brain. And I've listed up here some candidate diseases that could benefit from this technology. And you'll see here these are a list of different neurodegenerative diseases. And if you think about them, these are scary diseases to have in your loved ones because they are largely uncurable at this point. And so one of the approaches that we're addressing is how can we deliver genes to the brain so that we can try to treat some of these disorders. The promising part is that there has been quite a bit of research advances to understand the molecular mechanisms for these diseases. And so the theoretical drugs are known, and it's just really a matter of how can you get those drugs into the brain. So in terms of gene delivery to the brain, so this was that large plasmid that I showed you. That's about 1,000 nanometers in size. What we want to do is to get this actually not only inside the cell, but inside the nucleus of the cell, which is shown down here. And so the pathway for really large things to get inside the cell is first you have to condense it to a smaller size, typically less than 500 nanometers in diameter, so that it can be internalized into the cell. Large entities of this size are taken up into lipid encapsulated membranes called endosomes. And so the next critical part is for the material to get out of these endosomes and then to be able to deliver into the nucleus. So we learn a lot from biological mechanisms. So the viruses are very good at doing this. That's what happens when you get a viral infection, is the virus is able to deliver its nucleic acid into your cell. So let's look at one virus. This is the adenovirus, which causes respiratory illnesses. So the adenovirus has specific proteins that condense the DNA into its capsid, so inside the middle here. It has other proteins that allow it to recognize the cell surface, get taken in. Once it's inside the endosome, it's triggered by a pH drop to become membrane lytic so that it can get out of these lipid encapsulated vesicles. It can then hitchhike on highways inside the cell and get into the nucleus. So I'm showing here a very basic schematic of one of the materials that we've made. So understanding the molecular pathway that we have to take, we can then engineer materials that try to overcome these different steps. So what we have here is a single polymeric material. We have a nucleic acid binding domain that's shown in red. And that allows us to bind and condense the drug, in this case the gene. We have a particle shielding domain, because once we make these into particles, they have to be stable. We don't want them to bind to each other or to bind to off-target cells. So we have to actually keep them stable and relatively inert. And then we add on a cell targeting domain. So we add on, in our case, a peptide that helps it to recognize the cells that we want, in this case, neurons. And another important factor is that we have this degradable linkage, so that we can have a shell that will actually uncloak once the particle is inside the cell, and then allow for release of DNA. So then, combining all that, this is just a very rough schematic of the process, obviously not drawn to scale. So here are the particles. Again, we want them to be stable and inert when they're outside of the cell. But when it sees the neuron, we want it to be able to bind and recognize the receptors. It is then internalized into the cell. And the cell is actually a reducing environment. So we take advantage of that so that the reducing agents in the cell cytoplasm will then clip off and release this cloak that is around the particle, and then allow for the DNA to be released. So I have a talented polymer chemist in the lab, Hua, who came to us from China. So he synthesized what he calls his yin-yang agent. And so this allows us to make this type of block copolymer. It is a single molecule that's shown here that contains this internal disulfide bond. This is the bond that's cleaved once it's inside the cell. And he can do two types of polymerization. That's why he calls it the yin-yang double-headed agent. So he can polymerize one type of polymer off one end and a second type of polymer on the other. And so this polymer to the right, we can functionalize with the DNA binding agent. And this polymer to the left, we can then functionalize with a cell targeting ligand. And so that gives us the properties that we want. And what's really nice is with this type of synthesis, we have a high degree of control over the materials. So we can make the red, pink, and blue lengths very well controlled. And so we've been able to test a panel of these and optimize the structure for gene delivery. So let me just show you some of the results for this particular material. So again, the first step I told you is that the nucleic acid binding domains interact with DNA to form this particle. And we can visualize this by electron microscopy. We have the blue hydrophilic polymer that acts as a shield. This keeps the particles stable until they get inside the cell. So that's the particle shielding domain. And we can test this by doing a simple size stability assay. So we just put this in physiologic salt concentrations. And if we don't have the shield, that's the blue line, you can see that the particles aggregate and they start binding to each other and they get really large and usable. But if we have the shield on there, they're really stable up until we went out to 16 hours. I guess he went home after that. But it's stable for a long period of time. The third is that we put on this targeting ligand. And we've used a targeting ligand that's shown to bind to neurons. So he took this material and tested it in a neuron-like cell in our laboratory. And he shows that he can get some transfection with the non-targeted material, but he can increase the delivery efficiency by over 10-fold by putting on that ligand to recognize the neurons. And then finally, we have in the middle that linkage that allows the material to uncloak in the reducing environment of the cell. And he shows that here, this green line that I've now added into that particle sizing is when we take the core and shell materials and we add in a reducing agent, and we can see that with time, the coat is being shed and that the particles then start to aggregate. So with our collaborator in neurosurgery, we've taken these materials and injected them into the brains of mice. And as a test, we delivered a plasmid that codes for a gene from fireflies. So it's a luciferase gene, which then gives off light. So we basically monitor how much light is given off by the brains that we then collect. So we inject in the right brain, we collect the right brain. And so what we see here is these are the 4 different formulations. The first 2 are stable linkages that don't shed their shell. And the second 2 are sheddable shells. And then ones with the green, again, have the targeting ligand. And so what we can see is that in the mouse, we can get about 3-fold increase of delivery efficiency if we engineer in the cell targeting domain. And if we engineer a sheddable shell, we increase the delivery efficiency by about 6 to 8-fold. So overall, by combining these different attributes, we get about 15-fold increase over the baseline material. So The message that I wanted to get across in this first short story is that by understanding the molecular mechanisms of delivery, we can design functional materials that can be more effective in delivering for our specific application. And with our synthesis techniques, we've been able to make materials that are highly uniform and that can be scaled. So those are very important attributes for clinical translation and scale-up. And we've been able to use these materials both in cultured cell lines and also in animal models to show effective gene delivery. The second topic I wanted to talk about is drug delivery to tumors. And so the main message that I want to get across here is how do we achieve more specific delivery. So again, we're typically delivering toxic agents, and we want to get these more preferentially to the tumor cells and not to normal cells. There are two major approaches in getting more specific delivery. The first one is known as passive targeting. And to boil it down, what this takes advantage of is the size and the morphology of the delivery vehicle. So in this case, you're delivering material into the bloodstream. And if you get the particle size just right, you can get something that's large enough so that it doesn't come out in normal tissues. But the tumor tissue is quite different because the tumor is rapidly growing, and so the blood vessels are not very well formed there. And the blood vessels then become more leaky. So larger materials can actually exit the bloodstream in tumor blood vessels, whereas they're retained in normal blood vessels. So again, if you get the size just right, you get something that's too big to come out in normal blood vessels and hit normal cells. But when you get to the tumors, they're small enough that they can exit out of these leaky vasculature, and then you can get this accumulation at the tumor site. So this is called passive targeting because, again, it just takes advantage of these size restriction differences between tumor and normal tissue. The second example I'll talk to you about is active targeting. And in this case, we're taking advantage of specific molecular markers or proteins that are upregulated on tumor cells. And so tumors, for example, might upregulate certain receptors to take in more nutrients. And we can take advantage of that to get inside the tumors. And that's a very common approach as well. So in the first example, we have synthesized polymers that will self-assemble into particles. And we encapsulate within these particles small molecules. And so the two examples I'll give you today is a fluorescent dye, indocyanine green. And we use this just to track where things are going. This helps us in our design. And the active chemotherapy agent that we've used is Doxorubicin. And so this is a clinically used chemotherapy. So what we found, and this is actually in collaboration with a local company, Omeros, is that if we take these two polymers, which are very, very similar, so this PEO-PHE-PEO and a PEO-PPO-PEO, the only difference here is the center domain that's slightly different, but they're very similar polymers. But what we found is that if we mix them at different ratios, we can get morphologies of delivery vehicles that are very different. So if we take something that's 100% the second polymer, we get really tiny spheres that are about 10 nanometers in size. And if we start mixing in this first polymer, we start to transform those into filamentous structures. So 100% of this first polymer is really, really long filaments, and we have shorter filaments in these in-between sections. So we were really interested in this because other laboratories, like Dennis Discher's laboratory at UPenn, have shown that these different shapes will circulate for different amounts of times in the body. And so what we did was we tested this in tumor-bearing mice. So we inoculated tumors into these white mice. And in the first experiment, we just injected these different shapes containing fluorescent dye. And then we extracted the tumors after 24 hours. And what we could see is that at this particular mixture of polymers, the 7-to-3, we were getting quite a bit of increased tumor accumulation compared to these other formulations. And again, we believe that this is due to a passive targeting effect, where these things are leaking out at the tumor site and then accumulating in the tumor. If we take the same formulations and we encapsulate instead an anti-cancer agent, we can see some of the benefits of drug delivery. So here we're looking at tumor reduction, and we're delivering the active cancer agent. The black lines show the tumor growth when there's no treatment. The red line shows the tumor growth with the standard doxorubicin treatment. And the pink and green lines are two of our micelle formulations that worked with similar efficacy as doxorubicin. So you may wonder, why would this be any better? And that comes in the second graph here on the right-hand side, which shows the mouse body weight. So we also monitor the mouse body weight. And when the mouse is not feeling well, it does not eat well. And you can see that the mice that are treated with doxorubicin have a substantial drop in body weight. And that's attributed to the toxic effects of the drug. But you can see that at the same concentrations, the micelle formulations work just as well as a free drug, but in the absence of this large toxicity that results in the body weight loss. The second example I want to give you is for active targeting. So again, that's using a specific molecular interaction to direct things to the tumor. There's been a lot of work on targeting tumor cells. And in this case, I'm going to do something that's a little bit different. And I'm going to talk about cells that are in the tumor but that are not the cancer cells. And the cells that we're very interested in here are the macrophages that are in the tumors. So macrophages are cells of your immune system. They're part of the white blood cells that come in. And in the tumor environment, the macrophages are actually pro-tumor formation. So there's been several studies that have correlated that levels of macrophages also correlates with poor prognosis. So the more macrophages you have, the worse your outcome is for the cancer. And there's several possible reasons for this. The macrophages that are in the tumors, first, they will secrete angiogenic factors, which means they recruit blood vessel formation, which feed the tumors. The other things they do is they will recruit other white blood cells. And one of the cells that they recruit helps to have the tumor evade your immune system. The macrophages also promote tumor cell growth. So these are basically cells that help your tumor continue to grow. So the macrophages in this case, so we have lots of macrophages in our body, but the macrophages in this case are activated into what's known as an alternatively activated state, which is considered anti-inflammatory. There's other cases where these macrophages can be activated into different states. Now, if we want to target the macrophages in the tumors to help reduce tumor growth, we want to only target the tumor-associated macrophages, so just the macrophages that are in this anti-inflammatory state. We don't want to hurt the rest of the macrophages in the body. And when we thought about that, it's actually quite difficult to achieve that because even characterizing these cells at this point is quite difficult. Right? It takes a whole bunch of different surface markers to even figure out which kind of population you're working with. So we ended up using a phage display approach. So we got libraries of phage. So these are bacteria, virus that attack bacteria. And you can display on them over 2 billion different peptide sequences. And so we take this library of over 2 billion different sequences, and we try to select out the sequences that bind to our cells of interest. And so we do this using a panning approach where we take one type of macrophages that we're not interested in. We remove all the materials that bind to that population. We then take the remaining population and screen for the binders to our cells of interest. So in doing so, we've been able to find a peptide that binds very specifically to these anti-inflammatory macrophages that you can see here. And they bind very little to the pro-inflammatory macrophage and also to the other white blood cells that are in the body. So we're very excited about this because we think that this is one way that we can attack tumors from a different point of view. So instead of attacking the cancer cells, we're attacking the support cells as well. And we want to pursue this as a way to maybe potentiate the current anti-cancer therapies that are out there. So as a proof of concept, we show that we can take this peptide that we've identified and we attach a protein on. You can imagine, for example, attaching a toxin. In this case, we just attached a fluorescently labeled protein. And we now have cells that are the M2 are the anti-inflammatory macrophage. The M1 are the pro-inflammatory macrophage. And we've taken our peptide, labeled in green, and we've synthesized the same sequence but just in a scrambled order as a control. And what we can see is that the peptide that we've identified binds very specifically to these anti-inflammatory macrophage, but not to the pro-inflammatory macrophage, and that it's very sequence specific. So if we just switch up the amino acids, we no longer get any binding in any of the cell types. So that's an early stage project for us, but one that we're very excited about. So I'll summarize this section by saying that we are engineering materials so that we can get preferential delivery of drugs and imaging agents to tumors by taking advantage of the physical properties of these carriers. We are also taking advantage of differences in protein expression profiles in cells in the tumor for targeting. And we're particularly excited about the macrophage approach because these are the normal cells in your body. The tumor cells are genetically unstable, and so they mutate and they become resistant to drugs. But these macrophages are normal cells, and so you wouldn't expect them to mutate and become drug resistant. So that concludes my talk, but I wanted to thank the people that we've been working with. So the people in my laboratory are shown here in the light color. It's not very clear, I guess. But these are the lab members that have been involved in the projects that I presented today. The ones that are in the bright yellow are our collaborators, and we have collaborators both within the MOLES program, so Pat Staten and Tony Convertine. We collaborate very heavily with the medical school here at University of Washington. So we have collaborators in neurosurgery and then also pathology, as well as with the local industry. And much gratefulness to our funding support as well. So thank you very much for your attention.
David is probably most famous for developing the Rosetta software that allows you to actually predict protein structure and to do design on protein, which is the area where he's really focusing right now. And we're going to hear quite a bit about this. And this is extraordinary work, and we're very pleased to have him here. David.
All right, well, thanks, Francois, for that very nice introduction. And I just want to really say over and over again during my talk how absolutely ecstatic I am to be part of the Molecular Engineering Institute here. And I tried to, in my talk here, cover some of the, emphasize, I think, the overlaps and sort of the new areas that this institute will bring forth. We do molecular engineering. We're designing macromolecules, primarily proteins. And what I'm going to do today is first try and give you a quick view of how we go about doing this, then go through some of the particular areas we're working on, again, with trying to emphasize relations to the other aspects of molecular engineering at the UW. All right, so what's the basic idea? If you look in nature, there are millions of proteins which have these exquisite functions that evolved to solve the challenges that biology faced during evolution, the 4 billion years of evolution. And they do this in just incredibly well. That's why we're all able to function and think and do everything. Proteins are really the workhorses of living things. However, we face challenges now that were not faced during natural evolution. And for this reason, there may not be proteins which exist in nature to solve them. And so the question is, can we design a whole new world of synthetic proteins to address these 21st century challenges? All right, so how do we go about doing this? So proteins are these very long, complex polymers. They have the amazing property of folding to their lowest free energy state. So there's a big energy gap. There's a single folded state. And if we want to design proteins that fold up to structure new states, we need to be able to calculate the energies of different folded states reasonably accurately, and we need to be able to sample protein conformation space, which is very large, well enough to find this lowest energy state. That's, if we want to design proteins with new functions, we need hypotheses about what configurations of atoms would be necessary to achieve a desired function. So if we want to design an enzyme, a catalyst, and we need to hold, and we want it to have a certain shaped active site with certain types of catalytic functionalities, we need to be able to come up with an amino acid sequence which will fold up to a structure which holds those atoms in the right place. This is early days. These are complicated problems, and our methods really don't work very well. So we need to be able to test everything we make, and we need to be able to do that quickly and cheaply. Okay, so the first question is, how do we sample this huge space that's available to a polypeptide chain, which can have 100, 200, 300 amino acids and thousands of degrees of freedom? And the answer is we need coarse-grained sampling methods. This is a Monte Carlo simulation. We're trying to sample the degrees of freedom of the protein backbone as efficiently as possible. And this just shows a movie of a small protein folding up. It only takes about as much time as it's taken to show this movie. However, the space is very, very large. There are huge numbers of local minima, and so we need to carry out many, many independent such calculations. And we're fortunate to have a distributed computing project called Rosetta at home, so we're getting volunteers all around the world to help us carry out these calculations and find the global minima of the proteins that we're designing. So that's the problem of, given a sequence, predict the structure. But the protein design problem is, in a sense, the opposite. Suppose we have this structure, and we want to find a sequence which will fold up to it. So what we need to do is find a sequence which has very low free energy when it's in this structure. So here we need fast algorithms for iterating not over the possible shapes of the protein, but its chemical composition, that is, its amino acid sequence. So here we're trying to find an amino acid sequence which is very low in free energy when it has this structure. And so for all the applications I'm going to talk about for the rest of my talk, we're using a combination of these sampling methods and design methods to come up with sequences that fold up into the structures that carry out the functions that we hypothesize carry out functions that we are trying to engineer. All right, so the way this works is very simple in the abstract. So we start off with, suppose we want to make a new catalyst. We start off with a computer calculation of the optimal sequence for that desired structure or function, essentially using more complicated version of the two movies I just showed, just sort of show you kind of strung together. And then the beauty of modern biology, modern molecular biology, is then all we have to do is read off the amino acid sequence of the designed protein, because we know the amino acid sequence because the computer popped it out, and then we back translate it to a DNA sequence, and then you assemble it or you fill out a gene order form. And I am very excited because I've been doing this myself the last month, and I can tell you that I calculated a sequence which was supposed to fold up to a structure, and I read off the amino acid sequence, and I actually just emailed that amino acid sequence to a company, and they said, yes, we'll send it to you in a couple of weeks. And so I got the gene in last week, and I'm going to tell you later on what happened. But it's that easy. The manufacturing of DNA has really made this problem very much more tractable than it otherwise would be. Okay, so we're working on a lot of different areas. If you look in nature, proteins do all kinds of different things. So likewise, you might imagine we could try and make new proteins that do all sorts of different things. So catalysts, with Mary Lidstrom here, we have a project which is looking promising on coming up with a completely new CO2 fixation pathway. This is neat because it's Jay just gave a beautiful talk showing what you can do if you mix and match the enzymes found in nature. But there's some enzymes which don't exist in nature, and you can make them and then put together new pathways like that. I'm not going to talk about catalysis, though, because I don't have much time. I am going to talk about binding proteins, and in particular, the potential of binding proteins to be a new generation of therapeutics, where we can go in and design things that bind very specifically to pathogens, for example. I'm not going to talk about vaccines, but the basic idea is to make proteins that mimic epitopes on pathogens that will then trigger an immune response to them. I am going to talk about biomaterials because, again, I think there's a very nice overlap or synergy with other aspects of molecular engineering. And also, I'm trying to work on this myself, so I have to tell you about that. All right, so binding. The challenge is we have a target, which would be this protein that's shown as in the surface view here, and we want to design a binder to it. And we view this as sort of this interaction surface as a climber might view a climbing wall. How can we hold on to it? What are handholds and footholds? And what these stick-like objects are let's see if this works. Yeah, are just isolated amino acid side chains that have been docked onto the surface. So these are ways in which we can hold on to the surface. And then the next step is to design a protein which will hold all of these amino acid side chains in place to realize this to be able to bind this target. We've applied this in particular to the 1918 influenza virus. There are many different strains of influenza, and so it's a constant public health threat. And we've in particular been trying to aim to bind to a region of the virus down here. This is the viral coat protein here, which is very conserved among different strains. And so here's a blow-up of the groove on the virus that we're going to try and stick handholds and footholds into, or we're trying to stick hands and feet in. These are amino acid side chains. I'm not sure, I hope you can see this. Side chains sticking into the groove. And then the next step, as I said, was to try and connect these with a single polypeptide chain. And that's shown here. Here we've designed a small protein which holds these side chains into this groove in the virus. So this is the virus in the surface view here. And here's, as I said, our calculations are not perfect. Many of the proteins sequences that we come up to don't actually fold up to the structures we want. So we have to test many, many possibilities. But with modern DNA manufacturing, as I described, it's actually quite easy to do that. So we made on the order of 80 different designs, made genes for all of these, and then tested them. And here are two that actually bind the virus, which is shown in yellow. These are two different designs which bind it with quite high affinity. And you can see how they're holding these side chains up against the viral surface to make van der Waals hydrophobic interactions. And here's a hydrogen bond down here. We've been, with collaborators, we've been able to get crystal structures, be able to determine how these small molecules actually bind the virus. Here's the sorry, I'm losing my mouse. Here's the viral coat protein here. And here's how one of these is where it was supposed to be in our design model, and the other color is the actual crystal structure. So we've been able to, so what these structures show, this is for one of the two proteins I showed on the previous slide, and this is for the other, is that we've actually been able to design proteins which bind this target, and they really bind exactly as they were designed to do so. So that's important. Now, very excitingly, these proteins protect against viral infection in cell culture. They actually protect against almost all viruses in one of the two large families, group 1 influenza viruses, and we are now trying to make proteins that bind to class 2 viruses. And pharmaceutical company has licensed these and is trying to develop these as anti-influenza drugs. They're only 50 amino acids, so they're very small proteins. Okay, so the method is really, it's completely general. There's nothing that was special to influenza here. All we need to know is the structure of the target protein on the pathogen or whatever. And so this could be a general route to new drugs. But there's a problem. How do you get proteins in to attack proteins and targets inside the cell? We're very fortunate to be at the UW where Pat Staten and Suzie Pun, as you've heard, are working on delivery methods for getting proteins into cells, which is just a match made in heaven. So Pat gave me a couple slides illustrating a collaboration between his group and our group. He's developed methods for delivering proteins into cells. So we've designed a protein which is, it's actually shown on the next slide, which is supposed to, which blocks an anti-apoptotic pathway in lymphoma cancer cells. And he's basically taken this design protein which we made in very similar ways to that influenza binding protein, attached it to his polymer and delivery system, and when he puts it in, he and a student put it into cells, it reduces the viability of these cancer cells. So this is a very promising thing. I just wanted to show how there's synergy between protein delivery and protein design. Okay, so now the last part of my talk, I want to talk about designing new materials. And this slide just illustrates all the different types of materials you see in nature that proteins form. Actin filaments in our muscles, connective tissue, all of these things are made out of proteins. And so again, the question is, can we make new proteins that do new and useful things? So towards this end, we've been working on methods, so Nobu and Ryakoga in the group have been working on methods to design proteins of arbitrary shape. And they've gotten very good at this. And so we're able now to design proteins with quite a few different types of folds from scratch. And the next step is to figure out how to put these together. But first, I have to apologize, but I told you got my gene last week, and I was trying to make this crazy protein up on the top here, which is this ultra-long, skinny protein. There's nothing really like that that exists in nature. It's 12 nanometers long. And sitting next to me, Fabio and Posu were trying to make this other protein, which is 25 nanometers long. And we got the genes last week, and the pictures of them are up there. And the reason why you're having to see this, actually you can't probably see these images at all, but these are electron micrographs on the left and right of these fibers. And these proteins actually do form fibers of 25 and 12 nanometers long. So these you might think of as the struts for the materials of the future, where we can start assembling, if you imagine junctions, we can start putting these together with different angles and building all sorts of different types of nanostructures. So a protein origami, for those of you who are familiar with DNA origami. And in fact, we're about to order genes for putting these together to make right angles and boxes and things. So, so Neil King and others in the group are working for ways of assembling symmetric materials. And I'm just going to, I hope you can see this. This is a little bit better micrograph down here of a cube that Neil designed that's made up of 24 identical subunits. And you can see, I hope you can see the electron micrographs here, which look like little dice. This one, we've actually been able to get a crystal structure of, and the crystal structure looks exactly like the design model. And they're overlaid on the right here. But what's exciting about this is we're controlling, we can control the position of every atom in these things. We can change an amino acid in any of the proteins I've shown you and just make a new DNA sequence, and we have new functionality there. So for example, if we wanted to make rods coming out the corners of the cube, we would just put these two genes together. Or if we wanted to put a binding site for the flu, we can pick the flu binding proteins, and we're trying to put them on the corners of the cube so we get multivalent display. Neil's interested in putting drugs inside for drug delivery. There are really all kinds of things you can imagine doing with these type of materials. And I'm very interested in sort of more generally patterning. Like, so we saw pictures of solar cells this morning. Can we actually build things where, again, we can control these on the angstrom level, but get order over hundreds of nanometers? Can we make brand new types of materials for non-biological applications too? With Francois, there's a picture Francois sent me. So this is Frank DeMaio designed this. It's now a two-dimensional array, and Francois' student has pictures that look like this, which where the Fourier transform of this is consistent with the model, but we haven't quite pinned it down. But to show we can get 2D arrays, and so that's all I'm going to say about materials. But I think it's a very exciting area, and being in the Moley Institute, I think is just going to open up so many new opportunities for this. All right, so I wanted to close with just something kind of fun. I mentioned the Rosetta at Home project, where we're enlisting people's computer power from around the world to help actually do all of these problems. And so, for example, to test that my sequence, that whether it actually folded up to that structure that I was trying to design, I sent it out to the Rosetta at Home volunteers, and I was very, very happy 2 days later to see that the structure they predicted for was exactly the structure I had been trying to design that sequence to fold to. But we thought we could maybe go one step forward and get human brain power involved in these problems too. And this has been a really exciting collaboration with Zoran Popovic's group in the Center for Game Science here. And so in the past year, we've had some really exciting results with game players actually solving some hard problems that we had been kind of stuck on. And I'm just going to talk about the last one here because it's the one that has to do with design. So I told you we'd been making catalysts for some time. We designed a catalyst for the Diels-Alder reaction, which is an interesting reaction because there are no catalysts for this reaction in nature. But it wasn't a very good catalyst, and we were trying to figure out how we might make it better. It joins these two purple molecules, which I hope you can see on the left. So when you look at this design, this is the designed enzyme, which we had a structure of. there are two substrates which are shown here, and you can see that there's just a lot of open space here, whereas a real enzyme would come over and cover that space with amino acids and get more interactions. But it's not at all obvious how to do it. You can't just give this to a computer and say, fill that space. So instead, we gave it out to Foldit players, and Foldit's a competitive game. So then they were all competing with each other to see who could design loops that would make the most interactions with these two ligands. And so they could grow out different loops, and working together with an undergraduate student in the group, Chris Iben, they came up with this crazy insertions of two big helices, this purple bit here, and it gives a very striking increase in activity of this enzyme, making it very, very much better than it was before. And the thing that was even more amazing to me is that the crystal structure of this Foldit player designed enzyme is actually really close to what their design model was. So we can not only get the world involved in helping us by contributing computer power, but hopefully brain power as well. And we need it because these are hard problems. So a lot of people were involved in this work. Sorel and Tim and Aaron did the work on the hemagglutinin, the binding. Eric did the collaborative project with Pat Staten. Neil and Will and Frank have been working on the symmetric design. Fabio and Posu, I showed you their design. And then the games has been a collaboration with the Center for Game Science here at the UW. And then I'm really excited about all the collaborative possibilities with Molly, and absolutely really excited about being part of the program. So I'll stop there.